purines, and adenine, which is what ATPs contain, is an example of a purine. And the other ones are pyrimidines. Okay, so the examples here for the purines, either A or G. A stands for adenine. G stands for guanine. The pyrimidines, there's three of these. There's T's, U's, and C's. T's, U's, and C's are the pyrimidines. This is thymine, uracil, and cytidine. Nobody ever uses those words. It's really funny saying them. We don't use the letters, right? So when we talk about these things, we always refer to just the letters. Now, what we mean by purine is the following structure. The compound called purine, which is the basis for these things, looks like this. There's a 5-member ring and a 6-member ring fused together with nitrogens there, and there. This is purine. With alternating double and single bonds like that as purine. And pyrimidine looks like this. Just a single 6-member ring with 2 nitrogens. Like that. So this is purine. This is pyrimidine. And what differentiates amongst these things is just different substituents, which we'll come to those as we go along. So different stuff stuck on these different locations. More nitrogens or oxygens or whatever. Okay. Now, what makes something, so those are the bases, 5 different bases. Now, there's more than 5 bases, and we'll talk about those later on also. What makes something into a nucleoside is a base and a ribose. So, for example, adenosine has a base adenine. So this is adenosine. This is adenosine. So we've got an adenine here connected to the ribose sugar. So a base plus a sugar, always a ribose. Well, it could be a deoxyribose. Will be our nucleoside. Okay. And then a nucleotide will be a nucleoside with a phosphate. Now, the phosphate can be in any one of these positions on the ribose, but most commonly we'll see the phosphate here at the number 5 position. But it could be on the number 3 position as well. So either one of those will be. And we might have for a nucleotide. The nucleotide equivalent of adenosine, which is also known as adenosine. A nucleotide could have more than one phosphate. So if there were 2 phosphates, for example, it would be ADP. Now, adenosine is a synonym for AMP, so this is also known as AMP. But a nucleotide could have 2 phosphates. ADP, it could be 3 phosphates, that would be ATP. And that'll be true for all of the bases. Any of the bases can be in the nucleoside version or in the nucleotide version with 1, 2, 3 phosphates. Those are all the range of what you do. We eat these things in a normal diet that has DNA and RNA in it. When you eat those things, your body takes up the DNA and RNA, chops it up in the gut, like in the stage 1 of metabolism, into nucleotides. And nucleotide, the phosphate group is removed. And then in that form, they can be taken up in the bloodstream. But also, the sugar is also removed, and that's also taken up in the bloodstream as a base and the ribose. So most of the time, the bases that we need to construct DNA out of come from our diet most of the time. But not all the time, right? So there's two kinds of ways to be thinking about the synthesis of bases, right? And converting dietary bases into nucleic acids is called the salvage pathway. Salvage, a junkyard, which does salvage, that's the name of what a junkyard does in a formal sense. A salvage yard recycles old metal bits to make new metal bits, right? And so that's what we're doing here. So we're taking the detritus of our diet, right, and converting them into our own DNA. Or our body's cells may break down our own DNA and our own RNA to form bases, which we then reuse. Right? So that recycling within our bodies or recycling from another organism to us is called the salvage pathway. So it's simply making use of bases that's already there. And the other pathway is called de novo. If we don't get enough, and we need to make more for whatever reason, de novo means from scratch. So if you're going to make biscuits, you have a choice. You either get pop out of the can biscuits, which are already made by somebody else, and that would be your salvage pathway. Instead of throwing them away, you're making biscuits, right? Or you make them from scratch, right? So you buy the flour, the baking soda, and the milk, and all that stuff, and mix it all together, bake it yourself. So we have this choice if there's not enough bases in the diet.
So that's what we're doing in the salvage pathway. We're making bases in both of those pathways.
We're making bases here. We're recycling bases here. Right, so we don't have to go through the complicated
Recycling
process.
bases that were in our diet.
Either in our diet or broken down from just because they get broken down in our bodies naturally. Most of the time, almost everybody, most of the time is doing salvage pathway. You're usually, unless you're starving to death, you're not doing de novo.
So wouldn't de novo be the same thing if you're breaking down parts of your body to make?
No, de novo pathway, we're going to spend a little bit, we're going to do de novo next time. We might start it today. The de novo is taking individual carbons and individual nitrogens and piecing those individual atoms together, carbon and nitrogen by carbon and nitrogen, to make these structures. One by one by one by one. So de novo is like starting out with Lego blocks and just using the Legos to build a new thing. It's de novo, completely from scratch. Whereas the salvage pathway takes already formed bases and doesn't break them down. It uses the bases as bases. We do not do very much breaking down of bases. If we've got bases, we're going to use them. If we got more bases than what we need, we'll excrete them. We'll talk about that in a little bit. We'll excrete them more or less as bases. All right, so if there's bases there, we're going to use the salvage pathway. We don't have to make it fresh. But, under certain circumstances, we may have to make it fresh. So we can't use the de novo pathway. So we'll talk a little bit about this de novo. But keep in mind, most of the time we use salvage pathway. And so that's kind of going to be what I'm going to focus on, is salvage pathway. So let's do that right now. And the salvage pathway starts with the pentose phosphate pathway. In the pentose phosphate pathway, you take glucose and then you generate NADPH and, after several steps, ribose 5-phosphate. So that's the starting point for the ribose 5-phosphate. And then we take an ATP and add a pyrophosphate onto that. All right, now ribose 5-phosphate, the phosphate is at the number 5 position. At the number 1 position, we'll add 2 more phosphates. So let's do that. Ribose 5-phosphate, ribose 5-phosphate is the 2 extra phosphates going here. That to make phosphoribosylpyridine. pyrophosphate. ATP is involved, we generated AMP, and then the pyrophosphate component of the ATP, those two phosphates put wind up being put at the number one position right there. This goes by a short name, called PRPP for short, and we'll call it PRPP from here on. The enzyme is PRPP synthetase.
So what pathway is this? Were you telling us about? This is the salvage?
So I'm going to use this in the salvage pathway, but also the same enzyme is used in the de novo pathway. It's used in both pathways. So it's kind of a way, the committed step, the first step in the generation of all the nucleotides, no matter whether you're using the de novo or the other salvage, which is what we're doing right now.
So this is the salvage?
It's a salvage pathway. I'm focusing right now on salvage.
Okay.
I mean, it's very straightforward what happens next. We just take a base. So for example, here, I could add an adenine here, or one, anything I wanted to.
So there's a lot of energy in that right there, the phosphate?
Yeah, right. It's taking a lot of energy. That's an important question. I'm glad you asked it. The context in which our body is going to be making DNA is the context in which growth is permitted, right? Which means that there has to be adequate reserve. We talked about this the other day. We talked about, cannibalism and who survives and doesn't survive and all that sort of thing. If there are external resources abundant, which means that there will be abundant ATP